see my slides. Yeah, it's a pleasure to be here. Thanks for the invite, guys. It's a pleasure to be back. Yeah, so I'm just gonna go right into, what has been my project for the last 4 years now. I can't believe it's 4 years. So during my postdoc, similar to Liwan, I actually became very interested, even though I'm an immunoparasitology by training, in the skin as a barrier site, but not only as a barrier site, but also as the major sensory organ in the host, in the mammalian host. And the way this process operates, as you can see here, is mediated by a very, very diverse subsets of sensory neurons that innervate both the dermis and the epidermis. They can transmit different sensations. Some of these may be, noxious, others may be more unpleasant. In particular, I actually became interested in nociceptor neurons, or those neurons that actually are able to detect noxious stimuli, uncomfortable stimuli, such as pain or itch. Now, the reason why I became interested in these neurons is actually because of work from Isaac Chio's lab, Dan Kaplan, Ulrich von Adrian, Carol Sokol, and many, many others in the field nowadays, showing that these nociceptive neurons that again innervate the epidermis, they can actually directly react to different pathogens in the skin. Now, when you actually start scratching speaking of itching, scratching the literature on this, you'll actually see that the contributions of these sensory neurons are actually quite, they seem to be very context dependent. In the case of fungal infection with Candida, sensory neurons can actually recognize this pathogen and induce a pro-inflammatory response mediated by IL-17 to counteract these pathogens, but it's also detrimental in autoimmune diseases such as psoriasis. By contrast, Isaac's work actually showed that these neurons can be targeted by different bacterial species, and this interaction actually results in the downregulation of the skin immune responses, which were proposed or have been proposed to mediate bacterial dissemination. So basically, what this actually suggests is that sensory neurons are they almost function as a rheostat to either promote immune responses or actually reverse these, and these immune responses and the mechanisms that mediate these are actually not well understood. So inspired by all of these studies, I wanted to discuss with you today this concept that I've labeled sensory immunity. Sensory immunity basically refers to the idea, and Liwen again referred to it, that neurons can actively participate in the early detection of pathogens that target the skin, these barrier tissues. And the detection of these these pathogens is sent to the dorsal root ganglia next to the spinal cord, which is then later interpreted by the CNS as pain or itch. But also this prompt recognition of pathogens by sensory neurons is important to elicit a fast immune response at barrier tissues, again in the skin, to counteract the invasion by these pathogens. So with this idea in mind, one prediction that we can throw on this, if this idea is actually true, is that for a successful pathogen to invade the skin, it may have evolved ways to actually prevent the activation or the recognition by these sensory neurons that otherwise can rapidly induce an immune response against this invading pathogen. So this idea was great, but we wanted to test it. And in the Brodsky lab, we actually wanted to test it by using a parasite that may be familiar to some of you, which is Schistosoma mannsoni. Schistosoma mannsoni is a parasitic worm. It's a trematode that, along with other two species, Schistosoma hematobium and japonicum, infect about 1/4 of the global population, estimated by the WHO. And as you can see here in this video, I don't think I can play it again, but the larvae, the waterborne larvae, can rapidly be attracted to the skin, and then they invade the skin within minutes. It's estimated that for Schistosoma mannsoni, this process takes about 5 to 7 minutes. Now, these are large multicellular metazoan parasites. These are not, tiny bacteria. So you would imagine that this actual invasion, it's almost like alien, will induce a nice, inflammatory response. But surprisingly, people that get infected with Schistosoma mannsoni, they actually only develop these mild itching and mild inflammation responses, and this actually, sorry, I don't know how to do this. But this actually leads to the dissemination, to the invasion of the parasite, and the rapid dissemination throughout the body as they migrate through the dermis and rapidly find a blood vessel. So we took this parasite as a model organism to perhaps ask, well, is Schistosoma mannsoni actively manipulating or activating these itch-inducing neurons in order to facilitate its infection? And speaking of itch, I wanted to just touch base briefly that itch is mediated by very different or discrete subsets of sensory neurons in the skin. In mice, it's been described 3 different subsets of sensory neurons that are non-peptidic neurons, namely an MP1, MP2, and MP3, that can transmit itch in response to different chemicals. In particular, the ones I'm going to be talking about to you in this talk are the working. There you go. The MP2 neurons that are identified by the receptor MrgprA3 or A3 for short, and are the neurons that are able to transmit itch induced by the chemical chloroquine. Now, touching base a little bit on these neurons, again, they are defined by the receptor MrgprA3, which actually belongs to a very vast family of mass-related G protein coupled receptors. As I mentioned before, A3 is the primary receptor and actually identifies a very specific subset of sensory neurons that induce or transmit itch induced by the antimalarial drug chloroquine. And as you can see here, these A3 neurons, even though they do show some overlap with other TRPV1 expressing neurons, they actually constitute only 5% of the DRG neurons. And they do also have a human homologue, which is labeled by MrgprX1, suggesting that targeting these neurons may actually translate into therapeutic approaches. So we wanted to first ask if Smnsoni parasite can actually activate neurons that transmit this type of itch. And in order to do so, as Liwen mentioned, we use the mouse as an model organism, mostly because mice have a very well-defined behavior when it comes to itch, and this is defined in which we can evaluate the number of scratch bouts and the time that they spend scratching. A scratch bout being defined by the time that the mouse leaves its paw and starts scratching either the cheek or the dorsal skin, depending on where you apply your reagent, and then they return it to either the floor or they lick the paw. And this will be considered one bout. So using this system, we firstly tested if we inject mice with different treatments such as vehicle, PBS, 50 micrograms of the extract of the Cercari S. mannsoni or live Cercari, and we use chloroquine as a positive control for itch if mice exhibit any itch behavior. And as you can see here in these graphs, well, the vehicle treated mice, they show some spontaneous, that's expected, but really the injection, neither the injection with the live S. mannsoni Cercari or the extract caused a significant scratching compared to those mice treated with chloroquine, suggesting that in fact, injecting mice or exposing mice to S. mannsoni doesn't really induce itch. Now, if it doesn't induce itch, well, perhaps S. mannsoni can actually block itch. And to test that hypothesis, we designed an experiment in which mice were first treated with either vehicle or the antigen of S. mannsoni, that again doesn't seem to induce itch. And 1 hour later, those same mice were treated with chloroquine, and we evaluated the intensity of the itch response induced by chloroquine. And in this case, you guys can see here again, in the white boxes, those mice that were first treated with vehicle, they exhibit robust scratching and also scratching time. However, when those mice were pre-treated with the extract of S. mannsoni, it actually resulted in a significant reduction in the number of scratches and the time that these mice spent scratching, suggesting that a pre-exposure with Schistosoma mannsoni extract inhibit the ability of mice to respond to chloroquine and exhibit itch responses. Now, as I mentioned before, itch induced by chloroquine is specifically mediated by neurons expressing the receptor MrgprA3. So we decided to use an in vitro approach to specifically ask if A3 neurons can both see the S. mannsoni extract, and also if this first interaction altered their ability to respond to their cognate ligand chloroquine. So we generated DRG neuron cultures in vitro from MrgprA3 T tomato mice. And in these cultures, we're able to specifically identify A3 expressing neurons by their expression of T tomato, as you guys can see here. And then we loaded them with a calcium-sensitive Fura2 dye, which will allow us to track in real time the activity of neurons, just as Lewan just mentioned. Now, we took these cultures and treated them, first exposed them with either a vehicle or Esmansoni extract. And you guys have to remember that neurons typically respond very fast within minutes to the stimulus you give them. So we track the responses of individual A3 neurons for the first 5 minutes. As you guys can see here, when the neurons were exposed to vehicle, and in this graph, each line represents one cell that was gated, and we're tracking the calcium response to the activity of these neurons over time. We didn't really see any changes in the calcium influx or the neuronal activity when the neurons were exposed to vehicle. However, when neurons were first exposed to the schistosomatolite extract, we can see a couple of these neurons elevating their calcium influx, suggesting that in fact they can respond directly to the extract of Esmansoni parasites. Now, then we ask what will happen to these neurons if we expose them to their cognate ligand chloroquine. And as you guys can see here, while the vehicle exposed neurons, they can respond, they exhibit this rapid and robust influx of calcium, suggesting that they can indeed react to their cognate ligand. Those A3 neurons that were supposed to react to this chloroquine, they can no longer do so when they were pre-treated with the schistosomatolite Sarcari, suggesting that this first initial exposure may have prevented their ability to respond to chloroquine. And then just to make sure that these reduced responses are not due to cell death, we treat with 70 millimolar potassium chloride that should induce the depolarization of all neurons that are viable. And in both cases, both the vehicle and the Esmansoni extract treated neurons, they were still able to respond to potassium chloride, suggesting that in fact they are still viable. So what I've shown you so far is that schistosomatolite seems to be able to inhibit the activation of A3 neurons. So at this point of the project, we ask, why? Why would a parasite want to inhibit each neuron? Well, perhaps it's because these A3 neurons can actually, again, induce an immune response that counteracts the invasion of this parasite, as you guys can see here. So to test this hypothesis, we first thought, okay, so if these were true, what would be the ways that A3 neurons or each inducing neurons can actually prevent a parasitic infection? As I mentioned before, when this parasite infects the skin, it rapidly transforms into the next stage of the life cycle, the schistosomula, and then rapidly finds a blood vessel after going through the dermis and goes into the lung, a process in which might takes about 6 days. So if A3 neurons could actually induce immunity against schistosomatolite, perhaps it could be due to altering the epidermis and preventing the entry of Esmansoni Sarcari, repelling these parasites via some of these potential mechanisms, as you guys can see here. Or perhaps A3 neurons can actually induce a strong inflammatory response in the dermis that attacks and kills these parasites, preventing their migration into the lungs. So these were basically the two main ideas that we were thinking about when we designed the following experiments. So now to specifically target A3 neurons, we used a A3 Cree mice, and we crossed this with channel rosalind 2 mice, in which in these mice, the crossing with the Cree will eliminate this stop codon and allow the expression of channel rhodopsin 2, in this case, in A3 neurons. You may be asking what's channel rhodopsin 2? It's an artificial channel. It's actually derived from some algae that it's an adhesion channel that can be opened by exposure of blue light. So when mice are exposed in specific parts of the body, this cation channel opens in response to the blue light and allows the influx of cations, which will induce the depolarization of the membrane. Those calcium signals that you guys saw in response to chloroquine were mimicking this response by inducing blue light, specifically in those mice, in these mice in A3 neurons. So we took this approach, and basically we exposed mice in the ear, either control mice or A3 channel rhodopsin 2 mice, in daily sessions for 30 minutes to blue light in the ear. And after 5 days of stimulation, we exposed the ear of these mice to 500 S-Manzoni cori, and after 30 minutes, if you guys remember that video that I showed you earlier, this is more than enough time, should be more than enough time for those parasites to go through the skin and infect the mice. So we quantify the number of parasites that remain in the inoculum, therefore that couldn't go into the mouse, as a result of how resistant these mice may be to the parasite infection. And after 6 days, we also quantify the number of larvae that made it into the lungs to see if the activation of these neurons can actually either prevent the entry or the migration of the parasite into the mouse. And as you guys can see here, the pre-activation of the neurons here shown in the green group actually resulted in more parasites in the inoculum, meaning that they were unable to go into the skin. They were effectively repelled from the skin. And this also resulted in fewer parasites being able to reach the lungs, suggesting that activating A3 neurons with a gain-of-function approach induces resistance against parasitic infection with S-Manzoni. So this is fine, but the problem with a gain-of-function approach is that it may be quite artificial. So then we ask if these A3 neurons were actually required, if they participated during a regular S-Manzoni infection. So we generated A3-DTR mice, again, in which these A3 neurons now will express the diphtheria toxin receptor. And as you guys can see here, when we treat these mice with diphtheria toxin, specifically that is expressed, the receptor for this toxin is expressed in A3 neurons. This will induce or prevent the synthesis of proteins and induce cell death, again, specifically on A3 neurons. So then we ask if mice lost these A3 neurons, if this will affect the parasite burden that I just showed you. We did this by exposing either control or A3-DTR mice to diphtheria toxin for 3 weeks, and we repeated these assays. And surprisingly, the depletion of A3 neurons did not change did not make the mice more susceptible to the infection with S-Manzoni, which is actually not quite surprising if you think about it, because infection is already over 90-95% efficient. It's kind of hard to get better than that, in my opinion. But the depletion of A3 neurons actually resulted in more parasites reaching into the lungs, suggesting that these A3 neurons are also required to mount a proper immune response against the parasite. So then we went back to the literature and we asked what, if any, immune cells have been reported to interact with immune cells. And in the field, it's been recognized that very close to these sensory neurons, there are different immune cells in the vicinity of these, specifically myeloid cells. So then we asked if myeloid cells would actually reside in the vicinity of A3 neurons in the skin at baseline. To ask that question, we went back to the A3 T-tomato reporter mice, in which, as you guys can see in the in vitro approach, we can visualize the A3 neurons by their expression of T-tomato. Now, what I'm showing you here is a section of the skin, of the dorsal skin of these mice, and these are mice that have not been treated with anything. These are just mice in a cage. You can see here in red, these lines are actually the A3 nerve terminals. They innervate both the epidermis as well as the dermis. But surprisingly, in white, we stain for myeloid cells that express MHC class 2. And again, at baseline, we're able to find myeloid cells sitting very close to these A3 nerves, suggesting perhaps that when we activate or deplete these neurons, these myeloid cells may be affected. So based on this imaging, we went back to our optogenetic experiments, and we asked if macrophages, for example, were altered when we activated A3 neurons. As you guys can see here, in the case of the optogenetic mice, when we activate A3 neurons, we saw a significant increase in macrophages, both in percentages and numbers. And if we go to the loss of function approach, to the depletion of these neurons, depleting those A3 neurons also results in the reduction of macrophages in the skin, suggesting that perhaps macrophages mediate the protection against S-Manzoni induced by A3 neurons. And to ask that question, we specifically asked treated mice with either vehicle or clodonate-loaded liposomes. Clodonate will be engulfed by every phagocytic cell, and it will induce the killing of those phagocytic cells, such as macrophages. We treated mice specifically in the ear 3 times prior to starting the optogenetic protocol that I showed you before. And first, we confirmed that this strategy indeed depleted macrophages. Now you guys can see here by flow cytometry, when we evaluated the percentage as well as the numbers of macrophages in both the control and the optogenetic mice that were treated with fluoronate, this treatment indeed resulted in the profound reduction of macrophages within the skin. So if macrophages are depleted, then we ask if the protection against the parasite was altered in the absence of myeloid cells. And you guys can see here that depletion of macrophages in control mice didn't really alter the susceptibility of mice to Smith infection, but importantly, the protection that was induced by activating A3 neurons was reduced or was reversed when macrophages were depleted in this optogenetic system. So I can see that I'm going a little over time, so I'm rapidly just going to show how we got an idea of what's the mechanism behind this. We went back to, we performed a single-cell RNA experiment in the skin. We identified specifically 3 main clusters of myeloid cells, and these myeloid cells were enriched by with expression of these 3 genes, CRC, ANCALCA, and BAM1, which together they form the receptor for the neuropeptide CGRP, which is a 37 amino acid, a small peptide that is expressed by A3 neurons. So we then asked if 2 things. If myeloids can directly react to secretions such as CGRP by A3 neurons, and if CGRP mediates the activation of these myeloid cells by A3 neurons. So to ask that experiment, to ask that question, we culture A3 neurons. We stimulate with the A3 agonist chloroquine for 1 hour. We collected that supernatant and exposed wild-type macrophages, then just evaluated the expression of pro-inflammatory cytokines. And this, as you guys can see here, this exposure to neuron juice, if you may, soluble mediators from A3 neurons, actually resulted in their expression of pro-inflammatory cytokines. But importantly, if we repeat these experiments with RAM1 knockout endings that cannot respond to CGRP, we saw a significant reduction in the expression of pro-inflammatory cytokines induced by the A3 neuron supernatant. However, we still see some increases compared to vehicle, suggesting that CGRP is partially responsible for the effects of macrophages on myeloid cells. So just in the interest of time, I'm gonna stop there. So what I've shown you so far is that Esmanzona has the ability to inhibit A3 neurons, each inducing A3 neurons that would otherwise initiate a pro-inflammatory response in the skin, partially mediated by CGRP, that is able to not only prevent the infection with this parasite, but also prevent the dissemination of the parasite into the lungs. And I'm just going to skip a little bit again for the interest of time, but I just quickly want to acknowledge both my postdoc mentor, Lebrowski Herbert, as well as other members of the lab, specifically Camila Napri, our master's student, that was absolutely fundamental for the progress and the success of this project, as well as the other members of the Herbert lab, our collaborators in different institutions, and the funding that I've been granted over these years. And I'll be happy to take any questions.
Thank you, Dr. Rico. Is there any question anyone want to do or ask in the chat? Feel free to do it. And
and if not, we can go home.
So I do have a question. Thank you, Juan. It was really fantastic work. I really enjoy the way you present your data always. So I was wondering, how do the increase of macrophages that you see, it's because the neuron is promoting proliferation of the macrophages, or you see more recruitment?
That is an absolute great question. We did not formally test that in our study. However, Ulrich von Adrian has shown that NIV 1.8 neurons, which also includes some of the A3 neurons, when you activate these neurons, they secrete CCL2. And CCL2 will recruit CCR2 positive monocytes into the tissue. and then they will eventually mature into macrophages. So while we didn't specifically test it in our project, I would hypothesize that we're indeed inducing the recruitment of monocytes potentially that can mature into macrophages and also dendritic cells, because we also saw some increases in CEC2 dendritic cells.
And my second question, is there any possibility to use the extract of this parasite to like therapeutically, like to reduce itching in other contexts? Like I'm thinking about, for example, Lewin works like with in this case, bacteria.
Yeah, that is a great question, and I can tell you that's definitely a direction that we will be pursuing in my coming lab. I could not, be candid to say we can actually use the extract, because that's actually one of the major caveats of this study, that we use the extras. We don't really know what's in that extra, right? We don't, know if it's a protein, if it's a RNA or whatever you may. So we definitely have a lot of work to do to formally characterize this extract. I do have some candidates in mind. Schistosoma, secretes a lot of peptidases, similar to Staph aureus. So one possibility, especially if you go back to these calcium experiments, the fact that the inhibition happens within minutes would suggest to me that this is actually perhaps those proteases perhaps cleaving the receptor and preventing the neurons to be activated. So that will be one of those mechanisms. But again, we will have to define, and it would be great if, you can just put a label there, sell it, and,
Yeah.
Now, what's important about this, what I actually forgot to mention, that A3 neurons mediates non-histaminergic itch. So it's actually hard to sell new therapies for itch if you already have antihistamines. But guess what? There's a huge population that whose itch is not mediated by histamine that are left behind. So these kind of approaches that specifically target non-histaminergic itch neurons could be translated for those populations as well.
Wow. Thank you, Juan. Very, very interesting.
So, did you analyze other compartments or other cells that are involved into type 2 immune responses, like Th2 or Th9s?
Yeah, so you got me right there. So my heart, as you know, it's in type 2 immunity. I did my whole grad school, and listen, I went into this project, it was schistosoma. There's gotta be a type 2 component. Sadly to say, there wasn't. And if anything, when we activate A3 neurons, we saw a reduction on GATA3 expressing lymphocytes, specifically on Tregs, because Diane Mathis has shown very nicely that the Tregs in the skin, they actually express GATA3 at baseline. But we also saw a reduction on ILC2s when we activate these neurons. And consistent with this, we actually saw an increase on IL-17 expressing gamma delta T cells. So it really seems that when we activate A3 neurons, we skew the skin towards an IL-17 phenotype that blocks or reduces, type 2 immunity or ILC2s. Yeah, because we didn't really analyze a lot of Th2s because we don't really have a lot in the skin at baseline. So these mechanisms, and in the manuscript, we also show that gamma delta T cells are required for the protection, similar to macrophages. Gamma delta T cells are required. So it really seems that this is an IL-17 gamma delta T cell mediated mechanism.
So we have a question from Luis. So
go ahead.
Can you hear me?
Absolutely.
Yeah, I have one question. If can be possible that similar mechanism in the tuberculosis infection where the macrophages just can be activated, and I don't know if could be neurons that can be participate in the activation of these kind of cells in the lung, but without the proper activation, they can eliminate the infection. I don't know if a similar mechanism could be possible in that model.
Yeah, I mean, that is a fantastic question. And actually, that's something that I know some people in Isaac's group has actually addressed. I think Bottle a few years ago, I think and now they have the lab in, I think it's in St. Louis, Missouri. Yeah, but they were more focused on long innervating neurons. I'm not sure if they are doing it with TB, but I mean, listen, that is an absolutely fair question to ask. There are long innervating neurons. They are in the DRG, but there's also some vagal ganglia innervation. Whether this type of mechanisms can interact or occur in the pulmonary tissue can absolutely be possible. And I always say, like, when you do your IFA, that black space is in the mouse, it's really not black. It's full of nerves, extracellular matrix proteins, a lot of these things. So just because you see it black, like this kind of imaging, just because you see it black doesn't really mean it's empty. Like this whole black, so God knows what's in there. And at least and also Lewin studies show that those nerves that innervate these barrier tissues and modulate immune responses.
So he's thanking you in the chat, in case you
Oh.
are not able to see it. And finally, I wanted to ask you, did you analyze any changes in the macrophages? I mean, M1 or
Yeah.
M2?
So we did detect an increase in pro-inflammatory cytokines. I think it's mostly TNF. If we characterize like transcription factor, well, it's not really transcription, like INOS or ARCH1. I didn't really see changes in ARCH1, but then again, this actually goes back to, I don't really think this is like a type 2 mechanism. INOS, yeah, that INOS statement was a pain in the ass. I think it was up, but I was hesitant because I didn't really like the staining. But I can tell you those like monocytes actually stain better for INOS. Yeah, actually what didn't make it to the cut in the paper, but I'm working on another paper. After infection, you get a ton of neutrophils and ILC and monocytes, and those cells express INOS. So it's possible that similar to the monocytes, the macrophages are expressing INOS, and therefore they may have. I really don't like to have the M1 phenotype in vivo because I feel like that's very artificial. At least they may have some, INOS expressing signature, which actually may be good to address. With INOS, and it has been suggested to attack or to be a way that macrophages can attack ethnozonoid parasites. So that would make sense if they express INOS.
Okay, thank you. And I think that's it for today. And thank you everyone for being here. And I invite you to follow our social on Twitter, or what is the new one? Is blue or
Blue sky.
blue? Blue sky. So okay, thank you.
Thank you for the invite,
I guess
guys.
we'll see you next week.
All right, thank you, Liwan. Thank you, Juan.
Guys, thanks for inviting. Take care.
Bye.